So we have three fantastic speakers, and we will be dealing with pharmacology and the way how this can be, again, retinal diseases and other diseases treated. Philip Kiser was going to talk about visual cycle modulators, and I can tell you I a little know this fellow, and I think he's a next generation pharmacologist in ocular diseases and superstar in making.
Well, thank you, Chris, for that very generous introduction, and I thank the organizers for allowing me to participate in this very stimulating symposium, and it's a tough act to follow. Thank you, Dr. Demas, for the wonderful lecture. So, so the topic of my presentation is going to be molecular pharmacodynamics of visual cycle modulation, and I'll start by kind of giving a few a little bit of a backdrop about pharmacodynamics, drug mechanism of action. I'll go into a little bit of the history of the visual cycle before I get into the main story that I want to tell you. So whenever we're studying, for the antibodies that Dr. Demas talked about, these are very specific molecules, and I think we can really use the term specific in that case, that they're really binding to one other molecule to exert their effects probably. But whenever we're using small molecules, it's always important to realize that although it's convenient to talk about them having one target, in reality, they're probably going to have multiple targets, okay? And this goes for drugs too. That was used in the clinic, and we like to think about a drug having a particular mechanism of action, and that might actually be subject to change over time. And I just wanted to give a few examples of this that I'm kind of aware of. One of which is this drug called ranolazine, which is an anti-anginal therapy, and it was originally thought to be working as like a metabolic modulator, which was a very intriguing idea. And later we found out that this drug is probably actually exerting effects on ion channels, and that's how it's exerting efficacy in the case of angina. Another one that I really like is this drug gabapentin, which many of you are probably very familiar with, which is an anticonvulsant and an analgesic. And this was actually a rationally designed drug to try to mimic the endogenous molecule GABA, but to make it more hydrophobic so it could cross the blood-brain barrier. And it turns out actually that this molecule doesn't interact with the GABA receptor. It does something completely different. It's actually inhibiting calcium channels. Nevertheless, the drug is working. And so this kind of sets up the molecule that I'm going to talk about, which is this molecule is developed by Acusel, and it was based off of work really done in Chris Palczewski's lab, a molecule called amixistat, which again was a rationally designed drug that's trying to sort of mimic retinoids, in particular this putative transition state that's thought to be involved in the visual cycle pathway, which I'll talk about next. Okay. And this is just kind of to show you, and I'll kind of go over this again, just to kind of show you, if you look at these molecules separately, they may not have too much resemblance to your eye, but once you sort of overlay them, you can see kind of how one was derived from the other. So I'll give you a little bit about background about the visual cycle, and it's kind of interesting to talk about the history of this. So again, just to orient you, we're in the back of the eye here in the retina. And this is this study on this cycle, this visual cycle, which is really responsible for keeping our vision sustained over time. It started over 150 years ago by a couple of German physiologists, Bull and Kuhne. And it was Bull who made really this observation that whenever you take a retina out of an eye and you expose it to light, that it undergoes a series of color changes from this what was called visual purple to this sort of yellow color, and then finally to this sort of bleached state. And he's hypothesized that was really the basis for how we see things, that this bleaching initiated something that sent a message to the brain for us to see. And it was then this work was picked up by Kuhne, who made a really critical observation that if you take this bleached retina, put it back on the RPE, that it would give back this regenerated retina with the visual purple type color. And that kind of the chemistry of that was not really well understood for a long time until the work of George Wald in the 40s, 50s, 60s, where he was able to really resolve that this was due to the photochemical changes in a protein called rhodopsin, and the fact that it has a chromophore, 11-cis retinal chromophore that's bound to it that undergoes a photoisomerization. And he was also able to speculate that there was this system in the RPE that was responsible for regenerating this chromophore. And again, he won the Nobel Prize in Physiology and Medicine in 1967 for this work. And so it was just starts a movie. So now we understand in much greater detail There we go. Here we go. No keyboard, sorry. It's playing now, sorry. So we understand really how this is working now at really atomic level detail because of the work of Chris Palczewski and others, where light strikes this 11-cis retinal chromophore that causes a conformational change that leads to reorganization of the helices and rhodopsin, and that opens it up so that it's able to signal, okay, to the G protein. And we really now understand, again, thanks to the work of many investigators, how this chromophore, again, at the end of this process of this light activation, the pigment, this rhodopsin, loses its chromophore, and it's this apoopsin, and there has to be a system in place to regenerate it because this 11-cis is not spontaneously regenerated from all trans. And so this, there's a pathway in the back of the eye, again, that's, at least in the terms of rhodopsin, it's involving enzymes present in both the photoreceptors and the RPE. Many enzymes involved in this, but they're all basically working to convert a trans-retinoid back to an 11-cis retinoid. And the key protein that's of importance for this talk is a molecule called RP65. This is something that's been, was really identified by really 3 separate groups in 2005 as being the isomerase of this pathway after kind of a long history of some controversy what it was actually doing. And this has kind of been in the news recently, as you probably all know, that gene therapy for retinitis pigmentosa and LCA due to RP65 mutations has been approved by the FDA, and this is really the first gene therapy for an inherited disease. So it's kind of been, again, been mentioned quite a bit recently because of this. Okay, so RP65 is carrying out this key step of converting an all-trans retinal ester to 11-cis retinol. So it's the hallmark step of the visual cycle, making a trans retinoid into a cis retinoid. So this is all what I've described to you so far as physiological. This system is critical for maintaining our vision. If you don't have some of those enzymes, you will go blind. You'll have poor visual function, your retina will degenerate, and you'll be blind. Now there's another aspect to this visual cycle that is kind of a two-faced sort of situation where it's important physiologically, but because of the chemistry of this visual cycle involving these aldehydes, so retinaldehyde, 11-cis retinaldehyde, all-trans retinaldehyde, that these are reactive. So that's good because it's able to react covalently with rhodopsin to form this sort of inverse agonist state to keep things suppressed. So that's good. But this also confers sort of a promiscuous reactivity to other molecules, other nucleophiles in the cell. And it's thought that these can react to exert basically cellular toxicity. And so this can happen because of retinaldehyde reacting non-specifically to other proteins, and whenever it makes these adducts, it can make the protein non-functional, or it can also react with, for example, lipids to generate bisretinoids like A2E and retinal dimer, which are thought to be cellular toxins. Okay. And in the eye, these toxins are really thought to possibly be promoting certain types of eye disease, particularly Stargardt disease and AMD with geographic atrophy. And so if you look at these fundus autofluorescence images, what you can see are these areas in the central retina of dead cells, and they're surrounded by this area of hyperfluorescence. And in the eye, this hyperfluorescence is coming from lipofuscin, and these retinoids coming from the retinoid cycle are key components of that. And so the fact that this is surrounding these lesions, there's that idea that these bisretinoids are sort of promoting the growth of this geographic atrophy, and so therefore if you could slow down the production of lipofuscin, you could possibly spare the retina from further expansion of these lesions. And so whenever the RP65 knockout mouse was created in Mike Redmond's group, they made this sort of interesting observation that if you look in normal mice, and I should point out this lipofuscin is in probably all of our eyes, and it's not necessarily toxic, and it's present also in wild type mice. But if you knock out RP65, they don't accumulate lipofuscin, and they're very, very resistant to light-induced retinal degeneration. And so this led to the hypothesis that if we can somehow slow the development or the accumulation of this lipofuscin, that could be a therapy for AMD and Stargardt disease. And so this really the first, I would say, certainly the first molecule that was a really strong inhibitor of RP65 that could suppress the formation of lipofuscin and protect against light-induced retinal degeneration was a molecule called retinoamine, which was developed in Chris Palczewski's laboratory. And this is a molecule that was intended to mimic the putative transition state of the RP65 reaction, okay? It's thought to occur through this carbocation intermediate that's positively charged. And so the idea is if you put this primary amine, which would be protonated probably at least to some extent in physiological conditions, that would mimic that. It could bind to the active site of RP65 and really slow down the flux of the visual cycle. You have less rhodopsin, and so therefore you would have less lipofuscin being generated. And so this idea was really developed further, again, by Acusella. They developed this molecule, amixistat, that really was kind of stripped. The retinoid properties were sort of stripped away from this for reasons, retinoids tend to have bad side effects, and so they kind of wanted to get rid of this. And so they arrived at this molecule, amixistat. And so I'll mention that this molecule has been advanced quite a bit. It's actually already undergone phase 3 clinical trials for the treatment of age-related macular degeneration. And unfortunately for those patients, it did not show efficacy in that trial. And it's now being studied in Stargardt disease and diabetic retinopathy, potentially, where it may have really a better chance of showing efficacy. But part of why we really wanted to investigate this is to really understand the pharmacodynamics of this molecule, because we may be able to modify it so that possibly we could have efficacy, for example, in age-related macular degeneration. So we kind of started this study, and I'll just tell you, my original intent of studying this molecule didn't have a whole lot to do with translational research. It was, what I was hoping to do is use this molecule as a surrogate for a retinoid so I could understand how retinoids bound to the RP65 active site so I could understand catalysis. So that's really where it started, because we really didn't have a whole lot of luck using retinoids. Because they're too hydrophobic, we weren't able to get really high occupancy enzymes, ligand complexes for our structural studies. Okay, so this is a crystal structure that we determined of RP65 in complex with the amixistat. And it actually sort of serendipitously, we had it as sort of a ternary complex also with a fatty acid coordinated to the iron. And so that really was kind of provided the key additional evidence of how the retinoids would sit in the active site of RP65 during catalysis. And so it's positioned in this sort of proximal binding pocket next to the part of the molecule that interacts with the membrane. And yeah, the AI talks were sort of making me a little nervous. So this was experimentally determined. But fortunately for my livelihood, we can't really predict crystal structures and drug ligand complexes with sufficient accuracy. Yeah, so this is experimental, but I think I'm going to start looking more into AI approaches maybe for structure determination in the future, based off the talks this morning, which were very stimulating. This is kind of showing the interactions between these molecules in the active site. And I'll go into this in a little bit more detail, but it really resolved a lot, understood why this molecule could bind with high affinity to the RP65 active site. And so we wanted to kind of compare this molecule to its predecessor, retinolamine, to see how it compared in terms of suppressing the visual cycle, and both in vitro and in vivo. So this is an in vitro assay where we're measuring 11-cis retinol production by RP microsomes. And you can see that compared to retinolamine, amixistat, and I won't talk too much about this MB001 in this particular talk, but it's a derivative that we made that kind of restored some of the retinoid properties to the molecule. But amixistat's much more potent compared to retinolamine in this assay, and it was also much more potent in vivo. So this is a visual chromophore regeneration assay where we take a mice, shine light to bleach, basically all of the visual pigments, and then watch how much recovers after a period of time. You see very little recovery with amixistat and sort of an intermediate recovery with retinolamine. Okay. And so we have this much more potent effect on the visual cycle, and we wanted to kind of look and see what were the determinants of the molecule that allowed it to exert this very potent effect. And so what we saw here, the first thing is that amixistat is a chiral molecule. There's a possibility of R and S isomers at this position. And so we wanted, and I'll just mention that R was the one that was actually is being studied in clinical trials. And so it turns out actually that R is the most potent isomer, but not by a long shot. Whenever we crystallize RP65 with the racemic mixture, both R and S, what we observe is density just for the R. So it's kind of competing out the S. Now, we can actually crystallize the S isomer. What you can see is that the hydrogen bonding interactions are kind of less optimal for this particular molecule, and that's probably explains the difference in affinity. Okay, so what if the goal is to make a more potent RP65 inhibitor? Okay, so we've got this, this work done by Acusella wasn't really driven by any structural biology And so can we look at this structure and predict how could we make a more potent molecule? And if, actually going back here, so if we look at this part of the molecule, it's involved in a lot of electrostatic interactions that we didn't really think could be modified very easily. But the part of the molecule that looked like it might be more amenable to modification was this cyclohexyl ring, where we could see that there was these large sort of unexploited pockets on either side of it. And so we thought that if we made a molecule that could engage in these pockets, that might improve the binding affinity potentially. And so we made a few of these, and the one that seemed to work best was this molecule called, what we called MB004, where you can see that the IC50 was, it was about twice as potent or so as a mixostat. So that's good. We're kind of getting a handle on this as far as how you could boost the potency, and it's something that we're kind of continuing to look at, making substitutions at other sites of this molecule. We also wanted to look and see kind of like what the basis was for this really strong inhibition, and really what the duration of the inhibition was. And if we look, so this is, again, this is the visual chromophore regeneration assay that we that I explained to you previously. And a mixostat really suppresses visual cycle activity for a long time. This is up to 20 hours, you really don't see much recovery. And if you look even longer, out to 6 days, there's still very little recovery in the visual cycle activity, whereas retinolamine is starting to rebound a little bit. And so one of the things we knew from retinolamine was that it's actually a substrate for LRAT, and I didn't really point it out too much in the previous slide, but this is a molecule that can esterify retinol to make a retinal ester, and it can amidate molecules too that have primary amines. So we wanted to see if, like retinolamine, this molecule could be amidated, and in fact, it can. So this is a mass spec showing the free amixostat amide, and if we look in vivo, we can see that it's being formed. So this is part of what's going on. Amixostat's being distributing to the RPE, and it's being amidated, and it's sitting there and being slowly hydrolyzed to generate active molecule over a long period of time, leading to this prolonged suppression. Okay, so we also looked at one of the properties I mentioned of retinolamine is its ability to protect strongly against light-induced retinal degeneration. And for this study, where we're using again, a mouse model developed in the Palczewski lab, the ABCA4RDH8 double knockout mouse that's really more susceptible to light-induced degeneration. And so these are OCT images. You've seen some of these already today. If we look at, so really the part of this that we're quantifying is this area here called the outer nuclear layer. If you treat mice with vehicle, expose them to light, and look at the outer nuclear layer, it basically goes away. You just see this increased scattering here, and that's because the cells are dying. If you treat with a mixostat at 8 mg per kg or retinolamine at 8 mg per kg, you see this really nice preservation of the outer nuclear layer. Now, interestingly, despite really the higher potency of a mixostat, it wasn't able to protect at this lower dose, which was a little bit puzzling to us, and that's kind of quantified here. So that got us, again, thinking back to what we know about retinolamine and what it can do. Retinolamine is known to have not only the ability to bind to RP65 to inhibit its activity, but also to directly react with retinaldehyde, this toxin from the visual cycle that can form bisretinoids and cause other types of toxicity. So this was again a sort of an unexpected mechanism of action of retinolamine. So we asked the question: can a mixostat also directly react with retinaldehyde to form this type of an adduct here? And so we looked at this by first looking at it in vitro. Can we actually form this adduct? And indeed we can. This is mass spec data, and here's the molecular ion showing that it's actually formed. And then we treated mice with a mixostat, exposed them to light, and then extracted out the retinoids and see if we actually form the same compound in vivo. And indeed we have a peak here showing that it's forming with this characteristic selective reaction monitoring peak showing up here. So that's interesting. It looks like it's forming the Schiff base like retinolamine. And then the question is, well, it's doing that, but is that actually sort of an integral part of its protective effects against light damage? And this is where looking back at the crystal structure was helpful in helping us design a molecule that could allow us to address this question. So if you look at this primary amine, which is really essential for this retinaldehyde sequestration activity, it's involved in these sort of electrostatic interactions with the fatty acid and some other groups from the protein. And so we surmise that we might be able to replace that amine with another group capable of forming these electrostatic interactions, but one that's not able to react with retinal very well. And so to do this, we created this molecule called MB002, where we replaced the amine with a hydroxyl, which has poor retinaldehyde reactivity. And so we first tested this, again, visual chromophore regeneration assay. At a comparable dose to retinolamine, it achieved sort of the same type of suppression of the visual cycle. But unlike retinolamine, in this light damage model, you see that it was really not very able to protect against light damage relative to retinolamine. And this is actually, you'll note, at a much higher dose compared to what we used here. So there seems to be something going on here, that the suppression of the visual cycle per se is not the whole story, and that we really need this retinaldehyde sequestration activity in order to have the protective effects. And this is just, I just show this off briefly. Grajna will talk about this, but this is another way to evaluate the efficacy of these drugs by two-photon microscopy imaging, where you see if you have a mouse that's just given oil vehicle exposed to light, we can see our retinal pigment epithelium here in the photoreceptors, and these photoreceptors are starting to get big. They're fluorescent now, and they're swelling up, and they're going to die. In contrast to the no light control, where you don't really see much autofluorescence coming from the photoreceptor layer. Amixistat protects against this light damage. MB001 protects against it not very well at all, and that's quantified here, although there is some statistical decrease or some protection. The statistically significant is nowhere near what you can see with amixistat or the no light controls. Okay, and this, just real quickly, we proved that the molecule still binds to the RP65 active site. We did that by X-ray crystallography. And so then the idea comes, and I'll just say with amixistat, the problem with suppressing the visual cycle, as you might imagine, is that it suppresses your vision, right? And in patients who are taking this, they have problems with dark adaptation that's problematic. So amixistat, we've just shown you, has these two mechanisms of action, inhibiting the RP65 active site and forming a shift base. And really what we hypothesize is that by promoting this mechanism of action, we can have a protective compound without the side effects, okay? And so we did this by introducing a group onto amixistat that would be able to block its ability to interact with RP65, but yet maintain its ability to react with retinaldehyde. And we made several of these. I'm going to sort of show you kind of the winner from this particular round of screening, a molecule called MB005, where in vitro, it really has very little suppression of RP65 activity. In vivo, if you look at the visual chromophore regeneration, there's very little suppression at a 500 microgram dose with some suppression at a 1 milligram dose. We showed that like amixistat, it's able to form the shift-based conjugate. But despite the fact that it's not suppressing the visual cycle activity very much, we're still seeing relatively good protection against light-induced retinal degeneration, as shown with these OCT images. So we're on that sort of the right track. Clearly, there's a little bit of work to be done in terms of optimizing the structure to really limit even further how much these molecules can suppress the visual cycle, and that's something that we're continuing to work on now. Okay. So what I've shown you here is that these visuals, the structural biology, I hope I've convinced you that this can be used in sort of a translational approach, has allowed design of these visual cycle modulators with variable degrees of RP65 inhibition. Amixistat has two protective mechanisms of action. It can suppress the visual cycle directly, but it can also directly react with retinal to sequester it away to prevent it from reacting with molecules to generate toxicity. And so really, development of optimally effective visual cycle modulators is really going to require this careful balance between suppressing the visual cycle, trapping retinaldehyde, and also sort of controlling how great it is at being amidated and stored by LRAT, okay? Because whenever it's in its amidated form, it of course won't be able to exert its retinaldehyde sequestration activity. So we're going to have to find a good balance between those. So there's a lot of people to thank here. Chris Palczewski is the primary one who was really involved in the project conceptualization and design. Neil Peachy, a mentor on my career development award, who kind of taught me the basics of ERG. Experimentally, a lot of this was done by other people. So Jinya Zhang, who did a lot of the mouse work and also the mass spec, very talented person in Chris's lab, researcher, Chichen Dong, who did the OCT analysis, Grajna for the two-photon imaging, and Junzo Kinoshita for the ERG work. Our collaborators in chemistry who made the derivatives for us, X-ray crystallography, access to the beam lines, and then finally funding from Departments of Veterans Affairs and the National Eye Institute. Thank you for your attention. I'll be happy to take questions. Yeah, that's a good point. So in terms of, I think, so with your first question about SNPs and maybe the bottom line is there, I don't, to my knowledge, well, this is kind of recently changed now with a recent paper, but RP65 itself or LRAT have not really been linked, as far as I know, to susceptibility to developing age-related macular degeneration. Interestingly, there was a recent paper that came out from the Swarup lab that implicated CRBP, which is a binding protein in this visual cycle, as being involved in contributing to AMD, risk for AMD. As far as opsins and cone opsins, yeah, I'm not aware that there's anything, there's been any sort of genetic evidence that those are really modulating. I mean, at least for AMD in particular. I don't think those have shown up in the GWAS studies, but again, there's the one example of visual cycle with CRBP possibly be involved in modulating risk. Yes. Right, so I think, yeah, so I think that with the retinaldehyde sequestration, you're still going to be taking retinoid out of the cycle, essentially, but I think that that's going to be a much more transient thing, and it's not gonna, you're not gonna have this very prolonged suppression of the visual cycle that I think is causing the dark adaptation problem. So I mean, it's a great point in terms of you are still modulating the visual cycle. That's the reason why I didn't try to like rename these or whatever, because it's really the same thing, but I think that this activity is going to be more limited in duration, and that's gonna be less of a problem, because as you saw with the mice, these, even with one dose, their visual cycles staying kind of knocked out almost for like an entire week. And we hope that if you would just have retinaldehyde sequestration activity, that it would be a very much temporary effect, and they wouldn't have the dark adaptation problems. Thank you.